topic that I'm going to discuss today, of course, it's very evident from the title that we are decoding the language of cancer, for which we're using AI to reveal the hidden clones that are driving the treatment resistance. So, like Rupert said, I'm currently working as a postdoctoral fellow at Peter MacCallum Cancer Centre. And here we are heavily focused on leukemia, but this project is part of my PhD where it was heavily focused on AI and treating prostate cancer. So you will see a lot of influence of that into this slide. So I will begin with a quick introduction to what is single cell data. So a lot of people have been doing genomics analysis, and for a decade or so, people have been doing bulk sequencing. But the thing with bulk RNA-seq is, although it gives high sequencing depth, it takes the average of gene expression for the cells within a sample. So the heterogeneity within the tissue is completely masked. So that's a big problem in cancer, because in cancer we get heterogeneous clones, and our entire goal is to find those hidden and rare clones. So this can be solved using single cell RNA-seq data, which takes the gene expression of the individual cells, which defines all the heterogeneity that is often masked by the bulk RNA-seq data. So this single cell RNA-seq can play a key role in finding the tumor heterogeneity, especially the intra-tumor and the inter-tumor, like the heterogeneity in the tumor microenvironment. And we can explore all the diverse population of cancer cells that are present within the tumor. However, there are a couple of challenges with the single cell RNA-seq. That is, most of the single cell RNA-seq, like it was introduced last decade, which is not many years, like not even 10 years. So most of the single cell studies are still based on bulk RNA-seq, which was discovered way long back. And then the second problem is like it uses the marker genes from those bulk studies to characterize the heterogeneity. And then another problem with the single cell is that, like, since we are sequencing each of the cells, so we don't really have any clinical data that is associated with it, and also assessing the risks that are associated with each of the phenotype. Due to the complexity of the single cell data, it is very hard to find. So we asked a question, can we use AI to stratify the tumor risk based on the molecular phenotype without relying on any predefined marker genes? And also we can somehow transfer the knowledge that we have from the bulk studies and transfer it onto a single cell study. So for this, the first question that we ask, like, what if the cells could speak and what would their language tell us? So our assumption is that if we consider each of the cell as a sentence in a human language or a document, and each of the gene as a word within that documents or the word within that sentence. So basically this genes forms the vocabulary of all the cells that are present. And if we can note it down, then can we infer the cellular heterogeneity that is present there? So for this to transfer from a single cell data to AI, we use the natural language processing, which is a field of AI that is focused on the interaction between computer and human language. So basically, it's a comparison between the English language we speak and the language that cells speak. So here we have considered that the cells are analogous to the document or a sentence, and the genes are analogous to the words within those documents. So now, one of the major challenge was how do we bridge the gap between NLP data and the single cell data? So here we used a thing called term frequency matrix. So what this term frequency matrix is, which is used in NLP, that if I have two documents, document 1 and document 2, and let's say I have these words within those documents, so this term frequency matrix notes down the frequency of each of the words that are present in that document. So here you can see in document 1, I have 112, like IU jump, with a frequency of 112. It is similar in document 2, but the two sentences are very different when it comes to that. So we drew a parallel to single cell data where we have considered that each of the cell represents a document, and the expression of the genes is the frequency of that word or gene to be expressed in that cell. So we, under this assumption, our first question was, how do we find, just give me a second. Yeah. So our first question was, if we know that we have certain kind of documents present or cells present, and let's say each of these documents have some property, like if I'm drawing a parallel with the song, so let's say some of the documents have the lyrics of a jazz music, some of them are pop music, then can we group those documents based on their similarity? So we map that into the single cell data to identify the group of cells that exhibit very similar expression profile. So for doing that, we use the cell document analogy, where the cell is analogous to the document and the gene is analogous to a word. I'm kind of highlighting that thing because that is going to play a role in our model. So for this, we have used a doc2wake model as our underlying model, and we have modified our doc2wake model using transfer learning for further inference. So I have used two terms like doc2wake and transfer learning. So I will go a bit deep on what are those things. So doc2wake is an unsupervised document embedding model. So it works very similar. It is very similar to the concept of ChatGPT, where we can give multiple text input and it gives output of a numerical vector, which we call the embedding. So I will be using that term as well quite a lot. So this embedding is a numeric representation of a document. So our whole assumption behind using document is that just like a document, so a doc2wake can group two documents based on their semantic similarity. So if two documents are similar, and they have similar words expressed, then the cosine distance between those documents will be, between the embedding of those documents will be very less. That is, our doc2wake model can group those two documents which are very similar. Similarly, when we translate that to single cell, when we give like the cellular data into a doc2wake model, it can find the cells which are very similar. So the embeddings of two cells with similar gene expression pattern will be closer to each other. So now I'm coming to the transfer learning bit. So doc2wake doesn't inherently do transfer learning. It's not a property, like it doesn't do transfer learning. So that's not a property of doc2wake. But then we thought, like, there are huge atlases of single cell data which are already present. And this comes from the fact that in bulk RNA-seq, we have those marker genes and that we are overlaying on the single cell data. So we asked a question, like, if we are transferring the labels from bulk to the single, why can't we use the existing single cell data? So there are huge atlases like human cell atlas, mouse cell atlas. Why can't we overlay their information onto a single cell data which we need to characterize? So for that, we use the idea of transfer learning. So transfer learning is the property of an artificial neural network, where instead of training a neural network from scratch, we take a pre-trained model which has already been trained using a large source data and transfer its weight onto a smaller target data, which is our data of interest. So our assumption is that it is going to give us a better representation of the cell types within that data set. So now, so this is the overall workflow of SCellBOW. So I will go into a bit of details here. So at first, so here I'm taking one single cell data. So I have two types of data. One is the large source data for my pre-trained model, and then a smaller target data, which is my data set of interest, and I want to characterize it. So once I take the data set, I'm doing different layer of filtration. So first is the gene and the cell filtering. So basically in this step, I'm removing the genes which are not expressed above a certain threshold. And I'm also removing the spurious or the contaminated cells which is not giving any kind of result. So the next step is a log normalization once I'm done with the gene and the cell filtering. And then I took the highly variable genes from there. So why do I need the highly variable genes? So most of the single cell study, like even a single sample can give me up to 10,000 to 20,000. And then most of the studies, they kind of scale up to 5 to 6 uh, samples, if not uh, less. So once we have that, so basically we get really big data set, and the number of genes that are typically used in a single cell is 60,000. So let's say I have 60,000 cells and 60,000 genes. It makes the entire data set really big. So for that, I will only select the genes that are actually having that actually have some variability within the different treatment groups or biological phenotypes. So for that, I have selected the highly variable genes, and then I have done a Z-score scaling on the entire data set. So this is my pre-processing step, which even translate to the large source data set. So once I'm done with that, here I'm creating the bag of words. So basically, I am representing each of the gene expression matrix as a document. So let's say for cell 1, I have this document called cell 1 where I have multiplied the genes like based on its frequency. So let's say a gene A has a frequency of 2. So it will be represented twice within that document. So once it is done, I have shuffled it so that it is not skewed by highly expressed genes. So once I'm done with it, I have the document representation of the target data. Now I have done a similar thing on the using the source data. So my pre-processing step is exactly the same for the source data. And then I'm training a pre-trained model using this source data so that we can calibrate the weight. So basically the idea is I don't want to start from scratch training a neural network model. So if I already train a model where the weights are calibrated, then it can better train, like it when at the fine tuning stage, it can better represent the target data. So once I have my pre-trained model, which is trained on the large source data, I give this smaller target data as an input for fine tuning. So the output from this entire model are my embeddings for the target data sets, which are basically the numeric vectors. So once I have the numeric vector, I have applied Leiden clustering on top of it. And here I have represented it using the UMAP. So basically we are scaling down the entire dimension. So we started with 60,000. Then in the highly variable genes, we are scaling it down to, let's say, 3,000, and then we are taking it into an embedding space which has a vector size of 300, and then I'm representing it into a 2D space, so just 2 dimensions. So this is the overall architecture of SCellBOW clustering method. So now to check whether a model trained on one dataset can be used to better cluster cell types in another dataset, I have used 3 use cases using public dataset. I have also worked a bit on our own dataset, which was a collaboration with a group in France from University of Nantes, but I'm skipping that over because explaining that would take a lot of time. So I'm just sticking to the public data set and the analysis that I did here. So for this, I have used three use cases. So my first use case consists of the cells from normal prostate, where my source data has 1,20,000 cells, which is really big from benign prostate. And then I'm transfer learning onto 28K cells from normal prostate. So here, this use case gives us a lot of cells to work with. So we are checking whether our model is scalable or not. Then the next experiment is on a highly heterogeneous cells from peripheral blood of humans. So for the source, I'm using 68K PBMC data. So when I say 68K, this is the number of cells. So 68K cells from PBMC. And then for as a target, this data set has almost 3,000 cells from the same tissue. So here we are doing a fresh tissue to frozen tissue transfer learning for a very highly heterogeneous blood type. And then in the third use case, I have used a pancreas data set. So the whole idea behind is to see whether our model is robust to batch effect or not. So for this, I have used three data, like for the source, I have used three data set using different technologies. So one of the data set, as far as I remember, was sequenced using InDrop. Then the next one was Smarter. And then another was CellSec2. And then I'm transfer learning it onto 2K cells of a different technology. So here it is a SmartSeq data, which is a completely different technology. And my source data has multiple batches. So I'm seeing if those batch effect in the source data actually affect the target output. Because when we take a atlas level model for training, like data for training our any kind of model, it can cause some biasness because they come from different labs to make the atlas. So just to check that if our model is robust to that, I have used the pancreas data. Now to compare, so since SCellBOW is clustering, I have compared it to different standalone method. So there are two state-of-the-art method, which is ScanPy and SURET, which uses the graph theory and statistical method. And then there are a third tool, that is DESC. So this uses a deep learning approach for clustering the single cell, and Sphere, which uses a generative model to create this single cell clusters. And I have compared my data with across my model across different parameters. So the first is ARI, that is adjusted rand index. So it checks the accuracy of clustering. And then NMI, which so the second plot is for NMI, which checks how similar the predicted group is to the actual group, like the true cell type. And then silhouette index, which compares how well separated the different clusters are, that it calculates the intra-cluster versus intercluster distance for each of the groups that are there. So then there is another parameter, which is a resolution. So for resolution, we can say it is very similar to at what resolution we are taking a picture. So higher the resolution, that means we are getting more clusters. That is more clusters. They will like the cells are grouped into more number of clusters. So here I have taken a resolution of 0.2 to 2.0 at a step of 0.2. So for the normal prostate data, when I compared the ARI, it shows, we can see that ScanPy, SCellBOW outperforms rest of the tools, including ScanPy and SURET, which is like super famous and everyone uses it in the single cell field. Across all the resolution, it is performing better. And so for PBMC, like when the resolution is really low, like 0.2, you just get two clusters. Maybe it is outperforming, but then across the decent number of resolution, SCellBOW is really good. And same goes for the pancreas. We can see that it has highest ARI across all the resolution. Then we did the same experiment and we calculated the NMI instead of ARI. And here I did it across different resolution and took the average of it. So here you can see the median of the NMI is highest in case of normal prostate for SCellBOW. And then even for PBMC, and we can see the same trend in pancreas. And then when we calculate the silhouette index, that is the cell type in silhouette index, that is how well it can characterize the different cell type, we can see that SCellBOW has outperformed rest of the tools. Then since we have adapted our doc2wake model, the baseline model, into a transfer learning method, so we wanted to check if SCellBOW can outperform the existing transfer learning method. For that, we have used SCTM, which is a topic modeling tool. Then ITClus, it uses a deep autoencoder to find the clusters, and SCBERT, which uses a BERT model. So BERT is kind of, it came before the GPT. So these are the kind of first transformer models that were there. So one of the advantage of using SCellBOW is that compared to all these models, like all of them need the source annotation, that it is completely supervised. But then in case of SCellBOW, it is unsupervised. So when we compared SCellBOW for the normal prostate, PBMC, and pancreas, we saw the same trend for ARI, like it is highest across all the resolutions. And we saw the same trend in case of NMI as well. So this is very comparable to the accuracy. So here we were confident that SCellBOW has high accuracy. And when we compared the silhouette index, here we saw that SCellBOW has comparatively less silhouette index for the normal prostate data compared to other. So we were kind of wondering, like, what could be the cause? Like, I mean, it is giving less silhouette index, but does that mean it is giving worse clustering compared to other tools and they're better than ours? So here is the UMAP for that. So you can see this is my SCellBOW UMAP, then SCTM UMAP and the SCBERT. So they give higher silhouette index compared to SCellBOW. But from this plot, we can clearly see that the SCellBOW clusters are more localized compared to SCTM and SCBERT. In SCBERT, we can see it's just a single blob and it doesn't mean anything. Like when we overlay the cell types on top of it, sorry, I have removed that slide. I don't know why, of the cell type overlap. So it was completely mixed. It was just a single blob. So here when we plotted the alluvial plot to see the mapping of the different cell types into the cluster, this gave us confidence because we can see this basal epithelial cells, they are kind of going into individual cluster, and the SM here, the smooth muscle, is also going into a single cluster. Similar for fibroblast, it forms a single cluster. But when it comes to SCTM, we can see that all the clusters are very intermixed. Like we cannot find a single cluster which is well defined for some of this cell types like the fibroblast or the HELOC. And in case of SCBERT, it's the worst because we are not getting a pure cluster even for the basal epithelial. So seeing that SCellBOW is able to outperform so many tools that are already existing, we were confident that, okay, if somebody is using the transfer learning way of doing clustering, using a doc2wake model, they can actually get good clustering result and which can actually help to find the rare clones that are present. Then, as coming from a computer science background, one of the most important thing was to see how fast it is, because we don't want a tool that runs for days just to get an output. So we compared the SCellBOW with the different transfer learning tools using the pancreas dataset. So here we can see that ITClus takes 2 minutes to run when we have almost like 14K cells, which is fast because ITClus takes the intersection of genes between the source and the target dataset. So basically, we are losing some of the information and we saw really bad performance from ITClus. Then in case of SCellBOW, it's the next base in case of threading and multi-threading, where it takes 3 minutes without any thread, with multi-threading, and 8 minutes without using any threading. And SCATM takes like 3 times more than SCellBOW, although it had decent performance, uh, but not better than SCellBOW. It takes a bit longer. And we had the worst performance, like time-wise, for SCBIRD, which should be better than, like ideally, BERT is a later version of a Dr. model, but it took so long. So our hypothesis was like initializing the parameters is taking a lot of time for birds. So hence, it is taking 3 hours. So overall, SCellBOW gives us a good performance clustering wise, as well as it is fast and efficient to run. And anybody can run it in a commodity hardware. They don't need a GPU or anything. So I will move on to the next aim. Sorry. So until now, using the Doctorow model, all we have done is we have grouped the different kind of cells. So we have identified the clonal population that are there, and there are a couple of tools which does it. Like not couple, like there are more than 10 tools maybe existing which does the clustering, but none of the tools can identify. Like if we give those tools a tumor uh, cell data set, it can never identify like which clones are aggressive and which are not. So we tried to address that question where we asked a question like, can we differentiate the clones that have been identified using SCellBOW clustering and rank them based on their aggressiveness? So for this, we have developed a marker-free method which does that, called Phenotype Algebra, which is built on the SCellBOW embedding. So the whole idea of phenotype algebra is dependent on word algebra property of a Doctorow model, which finds the semantic similarities between the word embeddings based on its context. So for example, if I want to calculate the embedding of the queen, it would be very close to the of calculating, like subtracting the male from the embedding of the king and adding a female content to it. So we use this idea to design our phenotype algebra model, which does this addition and subtraction operations. So the whole negative operation is what it does is it calculates the level of aggressiveness when we subtract a particular subtype from the whole tumor, and the positive operation, it gives us the level of aggressiveness of a tumor when we subtract one or more clones from it in combination. So this is the whole workflow of phenotype algebra. So here we have taken the single cell data and the bulk RNA-seq data, which has some of the clinical data, that is the survival data attached to it. So once we have taken the single cell data, we have created the pseudo bulk based on each of the clones that we have identified. And then we have combined the pseudo bulk with the actual bulk, and we have used the Doctorow model onto it to get the embedding. So once we get the embedding, we have calculated the embedding of the whole tumor, because the way it works is it considers that we are calculating the negative selection. So what will be the aggressiveness once I drop a particular clone from the whole tumor? So for that, I have calculated, we have calculated the whole tumor. And once it is done, we have subtracted the embedding of each of the subtype to get a resultant embedding, which we have given as an input to a random survival model forest, which has been trained on the survival risk data from the bulk RNA-seq. And then we'll calculate using the regression that has been used in this random survival forest, like how our aggressiveness is being calculated. So I have the pseudo bulk for each of the single cell subtype, and it is calculating the aggressiveness of that based on the model that has been trained on the bulk embedding and the survival data. So I know it is a bit complicated, but it will be more clear once I give this example. So the first question now, like now we have moved into a completely in a cancer space. So the first question we asked is, can we use this tool to identify the intratumor heterogeneity. So for this, we have used the breast cancer data. So in breast cancer, we have five subtypes: luminal A, luminal B, HER2 and basal, and normal line. So luminal A and B is considered to be more on the normal side, whereas HER2 and basal is considered to be very aggressive in breast cancer. And normal, like, it is still considered as the artifact of sequencing, so they are called normal like because they are assumed as normal cells. But when we applied phenotype algebra on top of it, here you can see that it lines up really well for luminal A, HER2, luminal B, and basal. But somehow it gives normal like as the most aggressive type. So here it is a bit of a flip because we are subtracting the clone from the whole tumor and then calculating how the survivability improves. So lower the value of it is, like, lower the predicted risk score is, more aggressive that subtype is. So we found that like our result was showing that normal like is the most aggressive type, and we were a bit confused, like it should come out at the top because it is the normal cells. But then when we did a bit of literature digging, we found that, like, based on the current literature, this normal like subtype is actually an artifact of sorry, this normal like subtype has traces of Claudine Lowe's breast cancer, which is one of the most aggressive type of breast cancer that is being diagnosed right now. So then moving on to the next cancer, we asked a question if it can actually do the combination, like it does the positive operation. That is, if we can subtract more than two subtypes from the whole tumor. So for that, we have used the glioblastoma. So in glioblastoma, we have the proneural, which is more on the less aggressive side, whereas classical is the most aggressive, whereas MES kind of lies somewhere in the middle. So in this, we can see that we have the highest improval in survival when we subtract, a decrease in survival when we subtract the pro. And then it lines up like MES is next and then classical. And when we subtract two subtype in combination from the whole tumor, it gives us the best prognosis. So basically, we are subtracting the two most aggressive cancer subtype from the whole tumor, and it is giving us the best result. So this lines up as expected. And then I also check like whether we can subtract just two or if we can subtract more than two subtypes from the whole tumor. So for this, I have used a prostate cancer data set. So we have classified the whole prostate cancer into AR high, AR low, and neuroendocrine. So AR high is least aggressive, whereas neuroendocrine is the most aggressive type. And here also we can see it lines up perfectly. We have AR high, AR low, then neuroendocrine coming out to be the most aggressive. And when we subtracted all three of them, we saw the best improval in survival, meaning that we were able to successfully remove all the aggressive subtypes from the tumor. So what is left are all the benign cells within those single cell data set. But until now, all the results were in the cancer cells. So all the examples that I present are the known molecular subtypes of the cancer. So the next question that we asked, like, can we do it in a using phenotype algebra, can we identify the tumor cells from the tumor microenvironment cells, which are the immune cell, the stromal cells, as well as the extracellular matrix? And when we did that task, we saw that phenotype algebra was able to dissect that. So so this is my tumor, whole tumor. So on the left hand side, we can see all the subtypes that are present within the tumor of the cancer. And then on the right hand side of the tumor, we can see that it has all the normal cells, like the immune cells, the B cells, the T cells, the erythroids. So this is another application of this phenotype algebra module where we can use it to identify the cancerous cells from the non-cancerous cells. So here is a bit of, so until now I have shown the application of phenotype algebra and SCellBOW individually. So one is used for clustering and the second is for ranking those cluster. So here I will give, in interest of time, I will be really quick. So I will just show a couple of examples like where these tools can actually be applied. So the first application is for a prostate cancer data. So here I have combined SCellBOW for clustering the prostate cancer data. And then I have ranked it using phenotype algebra, like which clusters are the most aggressive type. So according to the literature in prostate cancer, we know that the neuroendocrine subtype, which is this particular cluster, is very aggressive subtype of prostate cancer, whereas the AR, like the AR subtype, is the least aggressive. Uh, so we found a cluster using SCellBOW, which comes out to be the most aggressive type. But our expectation was this neuroendocrine, which is cluster 4 lying here, should come out to be the most aggressive type. And this, like, one of the most weird thing was this is lying right in the middle of the AR cluster. So it shouldn't be, like, it should be the least aggressive. So anyways, we kind of dug up a bit more to find that whether this is the artifact of sequencing and coming from a single patient or a single site. We kind of proved here that no, it is coming from multiple patients. So that means it is present across ubiquitously across different patient, and it's not the artifact of sequencing. And also another property of this is it has traces of all the subtypes. So most of the other clusters, they are pure. So only one subtype is overlaying on that, but this particular cluster has the traces of neuroendocrine, AR high and AR low. Furthermore, when we did this gene set correlation analysis, we saw this particular cluster is highly involved in stemness, which shows that this is playing a role in the metastasis, as well as EMT, the epithelial to mesenchymal transition. And also this has high relevance, like this particular cluster is highly resistant to the drug. So all the pathways are coming up saying that, okay, this is a highly dedifferentiated phenotype of the prostate cancer. And then the angiogenesis and vasculature. So our assumption is that this is one of the most aggressive subtype of prostate cancer, which is yet to find. And then the next application for this could be in aiding the single cell annotation. So we know there are a lot of tools like SCTyper. Then I think there is a tool called CellID, which does this annotation based on the marker genes, and we kind of rely on them. But do they give the correct answer all the time? So how to check that? So we can use phenotype algebra model for that. So basically here what we have done is we have taken our own data set of prostate cancer where it gives us 5 cluster. So this is my tumor cluster. This is the, since this is the bone metastatic prostate cancer data, so these are the osteoblasts. And then this small cluster here, cluster 4, forms the immune cells. And then these are the non-viable cells that are there. So when we applied SCType, which is a very famous cell type annotation tool, we saw that most of the results kind of line up. But when it came to this particular cluster, it showed that this is a progenitor cluster. So these are most likely most of the progenitor cells. But when we applied phenotype algebra on top of it, we saw that this cluster is coming out to be highly aggressive. So, but progenitor cell is supposed to be the normal cells. They are not tumor cells. And here we are getting it as the most aggressive type. So when we did a bit of digging into it, what we found that this particular cluster 3, which is attached to our main tumor cluster, this has very similar gene expression pattern to the tumor cluster. So here it is evident that no, this is not a progenitor, this is rather a cancerous cluster. And since it was coming out to be most aggressive, we checked a couple of other things. So we saw that this cluster has very high gene expression, which is similar to our cancer, prostate cancer cluster. And then here we can also see that most of the cells in that particular cluster are involved in the cell cycling. So they are involved in the G2M phase. And they also express the G2M marker more compared to the normal prostate cancer cluster. So from this, we can see that when Phenotype Algebra lined up cluster 3 before cluster 0, which is our prostate cancer cluster, we can kind of deduce that, okay, this is a more aggressive prostate cancer subtype that we can detect from this data set. So this is the overall application. So summarizing everything, this AI, the SCellBOW and Phenotype Algebra, both of them are AI-based single-cell RNA-seq method, which can find the cellular heterogeneity and characterize those heterogeneity based on the aggressiveness. And here some of the key observation, like I presented, was it has high clustering accuracy, and the Phenotype Algebra module is able to order the different subtypes, different phenotypes that are present within tumor data based on their aggressiveness. And also using the SCellBOW and Phenotype Algebra in combination, we were able to find new subtypes, like I wouldn't call it novel subtypes, I would rather say novel clonal subpopulation within the prostate cancer data. And then these are some of the work that I was planning on doing when I was in TRI, that we can explore it, expand it to the bulk RNA-seq data, as well as we can do a multi-parametric thing for this entire data. So this tool has all, like this is currently published in eLife. So if anyone wants to go through the papers, so this is the title of the papers, but I think we have changed it in the latest version. So it's just artificial intelligence based tumor stratification. And if you want to try this tool in your tumor data set or normal data set, then you're most welcome to try it. And if you need any feedback for it, you can approach me and ask like how to use this, because this is using a transfer learning module. So it has few steps that we need to follow before we can actually get the correct results. So you're most welcome to do that. Last but not the least, I would really like to thank my supervisory team from QUT. So here, this is like the bottom 5. They are my supervisory team from QUT, and Dr. Dibaka is from IIIT Delhi. And thanks to Brett, Anya, and Melanie for their effort all through my PhD. And this is my current boss in Peter Mac, Mark Dawson, who gave me the opportunity to take all this work forward. But we are transitioning from prostate cancer to leukemia, which is lab specializes in, and all the collaborators who contributed with their data. So Jeremy and Antoine, they provided the spleen data, and Lisa, she provided me with the bone metastatic data, and definitely all the institute that made it possible. Thank you.